I want to talk about challenges and possible solutions in development of orally available peptide drugs. Peptide drugs have a lot of advantages over small molecular drugs, such as high affinity, high selectivity, low toxicity, and low risk of immunogenicity. But at the same time, they have very serious limitations. First of all, low metabolic stability in biological matrices and low oral bioavailability. Only few peptide drugs can be prescribed orally. And here in this table you can see all of them, not many. And even for them, oral bioavailability is disappointingly low, less than 1%. When our peptide drug enter to gastrointestinal tracts, it is considered as a food. So the main function of gastrointestinal tract is to digest our peptide to single amino acid so that these amino acids can be absorbed from small intestine to bloodstream. So before our peptide reaches their target organ or target tissue, it should survive in harsh needle of stomach with low pH and high level of all kinds of peptidases, and then go through mucus and through tight junction of enterocytes to reach bloodstream. And even then, our peptide can be digested by enzymes, plasma enzymes. So it should be stable enough against plasma enzymes. So if we want to develop the orally available peptide drug, we need to increase its stability in biological matrices and to increase their permeability from gastrointestinal lumen to the bloodstream. There are several approaches. How can we do it? First of all, we can modify amino acid sequence of our peptide. And usual method how to do it are capping of N-terminus and C-terminus using amidation, esterification, or acylation. Also, we may incorporate non-natural amino acid or even some heterocycles into the sequence. And also we can use cyclization of peptide, which not only serves to increase metabolic stability, but also to increase permeability. And this is success story. Desmopressin, analog of vasopressin, which was the first peptide drug used orally. And the success of this compound was achieved by dissemination of cysteine and replacement of A-arginine for D-arginine. And in this way, half-life time was increased, and also some formulation, addition of some formulation, allowed to develop this peptide as an orally available drug. In spite of success, bioavailability of this compound is still very low, 0.2%. Another approach to increase both metabolic stability and bioavailability is attachment of polyethylene glycol fragment to our peptide. And pegylation increases metabolic stability because it constrains the approach of enzyme to the site of cleavage, and while in plasma, it reduces renal clearance, renal washing out. And at the same time, PEG fragment possesses mucoadhesive properties, so it sticks to mucus and increases residence times of our peptide near tight junction. And in this way, the probability to go through tight junction increases as well. And here you can see how this approach was applied to create orally available insulin, Tregapil, and this compound is now in phase 2-3 clinical trials, and this insulin analog can be prescribed orally. Quite a similar approach, attachment of fatty acid. Fatty acid can increase metabolic stability the same way as PEC does. In addition, once in plasma, fatty acid can bind to albumin, and in this way, the peptide can be rescued from lysosomal degradation, and its half-life time can be increased. And there are two main ways to go through tight junction of enterocytes to bloodstream. First way is paracellular, so to go between cells. It is shown in the red line. And another way is transcellular. It is shown in green arrow, and so go through the cell. And it was shown that amino acids and peptides can usually use transcellular way. And transcellular way depends on lipophilicity of the molecule. Attachment of fatty acid increases lipophilicity of our peptide, and in this way it increases permeation through tight junction as well. Both PEG and fatty acid can be used not only as a part of the molecule, but as a part of formulation. And in this case, they can be called permeability enhancer. Besides PEG and fatty acids, there are several other classes of permeability enhancers with various mechanisms of action, but generally all of them open or destroy the integrity of tight junctions, lower mucus viscosity, and increase membrane fluidity. Several combinations of permeability enhancers have been developed and patented, and here you can see some of them. And again, success story, Rybelsus oral version of semaglutide, which is analog of GLP-1 peptide. It is known that GLP-1 peptide has half-life time only 2 minutes, and two approaches were applied to increase the half-life time of GLP-1. First one is attachment of fatty acid. It has been realized in liraglutide, and it increases circulation time up to 13 hours. And the second way is amino acid replacement. Alanine in the site of cleavage was replaced for amino isobutyric acid, and in this way half-life time was increased again up to 13 hours. And combination of these two findings led to semaglutide with half-life time 1 week. A selection of proper formulation containing permeability enhancer SNAC allowed to develop Rybelsus oral version of semaglutide. And though bioavailability of this compound is approximately 1%, so still very low, the long half-life time compensates this drawback, and Rybelsus showed very good results in clinical trials. One more idea, making peptides shorter. Natural peptide somatostatin possess very long half-life time, only 2 to 3 minutes, but its shorter analog with some amino acid replacements showed much higher half-life times. Here you can see on the slide, you can see some of analogs of somatostatin. And one of them, octreotide, was developed in orally available form. Though bioavailability of this compound still is very low. But I would like to emphasize that sometimes making peptides smaller increases their stability and permeability. Here in this slide you can see examples of ultra-short peptides, which mainly include dipeptides, tripeptides, their analogs and derivatives. And you can see that all of these peptides are prescribed orally. A small size of these ultra-short peptides makes them less susceptible for enzymatic degradation, and at the same time, it facilitates the penetration from intestine to bloodstream. But the question is, how can we develop ultra-short peptide which possesses the same activity as full-size peptide? I would like to demonstrate one of the possible approaches using GVS111 as an example. GVS111 is ethyl ester of phenylacetylproline glycine, and it has been developed as a memory enhancer. Now this compound is in the market in Russia and some other countries. But how we started to develop this compound? As a starting point, we took piracetam, marketed memory enhancer, which has been developed by Belgium company UCB. Even now, not very much is known about the mechanism of action of piracetam, but at the time when we started our study, almost nothing was known. And we put forward several very simple ideas. We hypothesized that piracetam interacts with some unknown endogenous receptor. And in this way, piracetam mimics active conformation of unknown endogenous ligand. And since most of endogenous ligands are peptides, we assumed that it could be modeled by some simple amino acid sequence. Considering structure of piracetam, we selected several key elements. Pyrrolidine ring, which is present in two amino acids, proline and pyroglutamic acids, and glycinamide, which is a part of almost any amino acid. So based on structural similarity and some molecular modeling, we designed two series of dipeptides, proline-containing dipeptides and pyroglutamyl-containing dipeptides. Among pyroglutamyl-containing dipeptides, the highest activity showed L-pyroglutamyl L-asparagine amide, and its effective dose was 3,000 times lower comparing with piracetam. Analyzing structure of natural peptides with similar activity, we found that pyroglutamyl asparagine sequence forms N-terminus of the main metabolite of arginine vasopressin. And it's known that both arginine vasopressin and its main metabolite show some effect on memory. A simple dipeptide containing proline glycinamide, didn't show any influence on the memory. But its molepophilic analog, cetyl ester of proline glycine, showed clear amnestic effect, so it worsened memory. Comparing structure of compounds which increase memory and decrease memory, we found that the main difference is presence or absence of carbonyl on pyrrolidine ring. And we assumed that this missing carbonyl could be introduced through acyl group. So we designed a series of acyl proline containing dipeptides. You can see in this table that many of these compounds showed effect on memory, and among them ethyl ester of phenylacetylproline glycine was chosen for further development. We called this compound GVS111. Based on structure-activity relationship, we assumed that GVS111 is a prodrug, and once that phenylacetyl group facilitates the penetration through blood-brain barrier, and once inside the brain, this compound converts to cycloproline glycine, which is a real drug. We confirmed our idea and observed formation of cycloproline glycine both in plasma and in brain. Later, we found in literature that cycloproline glycine is an endogenous compound which is formed during metabolism of insulin growth factor 1, and the level of cycloprolinglycins correlates with cognitive ability of humans. Now this compound is in the market, and it shows very good activity and very low toxicity. So its therapeutic window is 10,000. So very large therapeutic window. But at the same time, what is most important, this compound can be prescribed orally. So let's recall the strategy we used to develop GVS 111. We started from non-peptide drug pyracetam and tried to model its structure by simple amino acid sequence. And after some optimization, we developed this compound. But the question is, was it just pure luck, or we can use the same strategy to develop other ultra-short peptides? And the question is yes, this is a working strategy. Here is another compound, GZR123, which also have been developed in Moscow Institute of Pharmacology by my colleagues. And they started with non-peptide antipsychotic salperid. Considering the structure of salperid, one can again select pyrrolidine ring, which can be mimicked by proline or pyroglutamic acid, and some aromatic fragment, which can be modeled by tyrosine or phenylalanine. And a very simple dipeptide, proline tyrosineamide, showed some neuroleptic activity. Overlay of salperid and proline tyrosineamide showed quite a good correlation. Then they tried to find the same sequence, proline tyrosine, among peptides with similar activity, and very soon they found that proline tyrosine forms the beta term in main metabolite of angiotensin. After some modification, after some optimization, GZR123 have been developed, which is methyl ester of caproyl proline tyrosine. Now this compound is in clinical evaluation. One more example, also from Moscow Institute of Pharmacology. As a basic compound, is a starting point, alpiderm was chosen. And again, considering structure, one can select benzimidazole fragment, aromatic fragment, and some fragment containing alkyl chains. And this alpiderm was modeled by phenylalanine, tryptophan, leucine, or isoleucine. And after some optimization, two compounds, GD23 and GD102, has been developed, which showed quite good activity in animal models. To summarize, development of orally available peptides is a challenging goal. Two obstacles should be overcome. First of all, low stability in biological media and low penetration from gastrointestinal lumen to bloodstream. And to overcome these obstacles, several approaches can be used, such as modification of amino acid sequence, including PEG or fatty acid fragments into the structure of the molecule, using some permeability enhancers, and shortening of amino acid sequence. And I illustrated one of the approaches which can be used to create orally available ultra-short peptide. And this approach has been illustrated by the development of GVS 111 dipeptide derivatives with cognitive enhancer properties.